Good morning, dear students, and welcome back. I am Dr. Savita Tidame, and I am going to teach you practical, that is zoology paper 3, and with the paper code Zo-113. So, in this online practical series, today we'll be studying the practical. That is, the aim of our practical is the study of a microscopic fauna of freshwater ecosystem, especially from the pond, right? So study of microscopic fauna. So whatever means, if you see a water or if you take a pond water, so pond water is having number of planktons in it, right? Number of planktons are present, microscopic animals, microscopic plants are there. So all together, that is microscopic plants and microscopic animals all together comprises the planktons, and they are further divided into phytoplanktons and zooplanktons, right? They are further divided into phytoplanktons and zooplanktons. So to whom we can call as a zooplanktons or phytoplanktons, right? So the phytoplanktons are the primary producers. Phytos stand for the plant origin, or they bears their own food material, they bears their chlorophyll pigment. With the help of that, in present of sunlight, these small microscopic plants, they can synthesize their food material. So phytoplanktons are considered to be the primary producers, whereas those microscopic organisms, they don't have presence of a chlorophyll, or they are not able to synthesize their own food material, right? Rather, they are movable. They can move from one place to another place. They can migrate, right? And such organisms, they consider to be the zooplanktons, and the zooplanktons are nothing but primary link in the food chain, right? Because see, phytoplanktons are there, which are the producers, unicellular phytoplanktons, microscopic, right, on which the zooplanktons they used to feed, on the smaller zooplanktons, larger zooplanktons they used to feed, on which again crustacean, many other small fishes they used to feed, and on the small fishes, larger fishes they used to feed. So the zooplanktons and phytoplanktons, they are important component of the food chain, right? That you might have studied in the first semester, that is animal ecology subject, right? In the ecology, you might have studied this, the food chain and food web. So this phytoplanktons and zooplanktons are the important component of the food chain, and if an ecosystem that bears a ample or a good amount of zooplankton or a phytoplankton, so that means a healthy ecosystem. That is the indication of the healthy ecosystem, right? Okay, so it means whatever water we drink, that should have ample amount of zooplankton. Is it like that? No, it's not like that, because whatever water we drink, that is come through the processing, right? So chlorine is added, and it has been purified further, so that we can get a pure water without such a planktons. So list very negligible, we can say, if one or two is present, right? Okay. So the any ecosystem, just like we said, a pond ecosystem, right? Okay, so dear students, let me show you the diagram of pond ecosystem. So they say this is a pond ecosystem. You can see a small pond is there, right? Size is again variable, but it has been bounded from all the side, right? Having a depth that is 12 to 15 feet depth. Okay, fine. So in this pond ecosystem, if you see here, phytoplanktons are there, which can be easily seen, but along with the phytoplanktons, number of small zooplanktons and microscopic phytoplanktons are there. These are nothing but the submerged aquatic vegetation, submerged plants, right? They are not considered to be the microscopic plants or phytoplanktons, though they are the phytoplanktons but larger sized. We are talking about microscopic phytoplanktons, right? and zooplankton. So all they are present here in this ecosystem, pond ecosystem, and on that small microscopic phytoplankton, small microscopic zooplanktons they used to feed, on which the other crustaceans, small fishes they used to feed, on which larger fishes they used to feed, right? And so these aquatic birds, and they also feeding on the fishes. Human is also feeding on the fishes. So the larger animals, they are depend on the smaller ones. So similarly, the phytoplanktons and zooplanktons, especially, they are playing very important and crucial role for maintenance of the ecosystem. If the primary producers are not there, then how these larger organisms they can feed? They cannot get their food material, right? They cannot get the food material, so ultimately they can't feed, right? So the planktons, that is zooplanktons, are an important link in the freshwater habitat, or you can say in the food chain, right? So with the help of these pictures, you might have got the idea about how the pond ecosystem is there, why the zooplankton, phytoplanktons are important, and how they act as a connecting link, or they act as a link in the food chain or food web. Now let's go back to the study of microscopic fauna, right? So after introduction, okay, what is present in this? Number of zooplanktons are there. Now zooplanktons is not single animal to which we can refer it as the zooplankton. No, number of microscopic organisms are there, right? They called as a zooplankton. See, I have listed here: rotifers, copepods, protozoans, Daphnia, and ostracoda are nothing but the groups of zooplankton. It doesn't mean the rotifer is a single organism. Huge number, hundreds, thousands of organisms comes under the rotifera. Means hundreds of animals belongs to copepods. Similarly, the protozoans, again a vast group. Daphnia, that is a cladocera organism. Ostracods are also there, but we have listed them into a single group, just like we human, right? Human, huge number of humans are there, right? Everybody is having a different name, right? I also having a specific name, that is Dr. Savita Tidame, right? You have your own name, but we all together considered as a what? Grouped as a human being, Homo sapiens, isn't it? Similarly, these organisms are also grouped into a specific group or a specific phylum or order or a class, right? So rotifers, copepods, protozoans, Daphnia, ostracoda are present into the freshwater as a zooplankton. Now let's say, for performing this experiment, what do we need? Study of microscopic fauna of the freshwater ecosystem. Requirements: we need a compound microscope, well working and clean compound microscope we need a dropper to take a water sample. On the slide we need a glycerine that prevent the hardening of the planktons present in the water, and most of all, most important, we need a water sample, pond water sample, because without a pond water sample, how do we conduct our experiment? We can't conduct it, right? So we need these things, that is compound microscope, dropper, glycerin, and pond. Okay, now let's see what is the procedure, how we can conduct this practical. So materials and method, first of all we'll see. So for the study of zooplankton, pond water sample were collected from the college botanical garden. For the practical, performing this practical, we always collect a pond water from our botanical garden, because it is very rich water containing huge number of zooplanktons in it. So we take that water. No doubt, you can take any pond water, right? But if the water is having less vegetation, so you'll get less amount of planktons in it. Okay, so the zooplanktons were identified qualitatively, means on the basis of their numbers. What are the different types of, like rotifers, copepods, cladocera, like that. So they are identified qualitatively under the microscope using drop count method, and the detailed procedure is given here. Drop count method means what? We have to count the water sample or the planktons drop by drop, right? Okay, so procedure is given here. The beaker containing water sample is already given to you, right? Say, just imagine we are performing this experiment and I am dictating you the procedure. Okay. Take a beaker, or the beaker with the water sample, pond water sample is already given to you, and how you will perform the experiment. So first of all, you have to take a clean glass slide, and on that glass slide put a drop of a pond water including some phytoplankton, right? So you have to take a drop of a pond water sample including little bit of a phytoplankton and put it on a clean glass slide. Then after that drop, you have to put a drop of a glycerin on it, so that the glycerin will prevent desiccation of the organism or desiccation of the planktons present in it, right? So glycerin is added. You cannot add a glycerin, this is optional one. So after adding a glycerin, you can put a cover glass on the drop so that it will remain as it is for a long time and you can easily observe, right? Or you may not add, or you may not put a cover glass, that is again optional one. Okay, so after putting a cover glass, adjust your microscope first of all at the 10x magnification, because 10x magnification and 10x or 15x magnification of your eyepiece is going to give you 100 to 150 times magnification of the image. So 10x magnification is sufficient, no need to adjust it on the 45x, right? So 10x magnification of the objective is sufficient, and try to find out the different zooplankton. You have to put your slide under 10x magnification, move your slide so as to locate the zooplankton present in it. While observing, you should not start with the center point of the drop. You have to start it from one point of the drop. So start from one end of the drop and with a zigzag movement, that is, if you go from one end to another end, again slightly move your slide, again go back to the opposite side, again slightly move your slide, again go back to opposite side. So like this you have to observe your slide in a zigzag manner. Why this is done? Because we cannot, or we don't want to miss any field. So you can get easily all means whatever amount of planktons present in zooplanktons present in the drop, you can easily count them all. No doubt this is a live sample, so the zooplanktons keep on moving, but for preservation we also add 10% formalin to the sample. But during this preservation, all the zooplanktons they get killed, and in that again you can easily observe. So that is also another part of the procedure. So you will find the zooplanktons, okay, in the drop, including protozoans. You may find protozoans, you may find rotifers, cyclops or daphnia in it. So what you will do? So you have to draw a rough sketch of the plankton present in it, right? Identify it with the help of your teacher and reference given to you. So you, as soon as you observe the organism, if you are able to identify it by means of yourself, you have to note it down, or if you are not able to identify it, you can call your teacher, right? And then you'll get the specimen identified. So follow the procedure for 5 to 6 times to get a large number of planktons. Why we have to follow this procedure for number of times? Because if you take one drop, you will get maybe 4 or 5 animals, 4 or 5 zooplanktons in it, right? Is it sufficient to show the plankton community, or yes, plankton community of a particular pond? No, you cannot say like this. So at least you have to count 10 to 15 drops from each water sample. But as in the practical, 10 to 15 drops will take, counting of a 10 to 15 drops will take a long time. So instead we'll do, or we'll repeat this procedure for 5 to 6 times, right? So you have to discard your drop, again take a fresh water drop, again observe it, follow the procedure, observe it, note down the zooplankton present in it, again discard it. So this procedure is going to be repeated again and again for 5 to 6 times. So here observation and result, but before observation and result, what we can do? The diagrammatic representation of the procedure what we studied up till now. So take a drop of a water sample, after that observe under 10x magnification. After that, identify the zooplanktons. Zooplanktons like this you can observe in the field. So whatever field you are observing, you can observe the planktons like this. So this is nothing but a cyclops. This is also cyclops, right? This is Daphnia, that is a cladocera. This is also Daphnia, right? So such a kind of animals you will identify, you will get under the field, right? Again, you have to change the field. You have to count them. Again, move away from the field so that you can get more organism, different types of organism or planktons in it, right? So change the field, identify and note the type of zooplankton present in it. After that, what is the next step? Discard the drop. When you observe, when you move completely the drop and you have to discard it, then clean your slide and again take a drop of a water sample on it. Again follow the procedure. So this procedure, entire procedure, you have to repeat for how many times? For 5 to 6 times, so that you can get more and more different types of planktons, especially zooplanktons in it. Right? Okay. Now let's move ahead and see what is the observation and result. So in the given water sample, following types of zooplanktons were observed. So you will observe number of zooplanktons, but we have categorized them into different groups and with a little bit of a characteristic how we can able to identify the plankton, right? So here I have given the picture of the representative group of that specific type as well as few characteristics. So you will write all this, that is you have to draw a diagram, rough diagram and follow this writing. You have to write this in your journal, right? Okay. So the cyclops, if such a structure is observed, there is a maybe modification or a different type of cyclops or a copepores you can observe, but only you have to remember if two antennae is there, one is short and one is long. So this is again general characteristic observed, right? Body is what? Elongated. At the broader head side, that is a metacephalon, and posterior side is what? Narrow one. Entire body is segmented, right? So the urosome or you can say anterior body is broader one, having a single carapace or having a single segment, right? Here you can see a single eye, presence of a single eye is again one of the characteristic feature. Two pairs of antennae is a characteristic feature, right? Again, along with that, there females, they carries egg sac or egg pouches. So these are nothing but the egg sac or egg pouches which are attached to the body of a female, right? So the copepores, they are having forked tail-like structure. Okay, they called as a furcal rami and which are having presence of a CT-like structure. So generalized structure of the cyclops or copepoda is given here in this diagram, and if the animal or under the field, if you can see such a kind of animal, you have to consider or you have to group it into a cyclops or a copepode, right? Cyclops is one of the type of a copepod, but which is most generally observed. So we have said here cyclops. Okay, then if you have changed a field and you got this another type of organism that is called as a rotifer, right? No doubt this is not the only one type of a rotifer, but varieties of different types of rotifers you can observe. Some of them, they are like this elongated body, some of them are having a rounded body structure, some of them having a dumbbell shape, not exactly, but like a funnel shaped structure, right? So different types of a rotifers specially present in the water, and we have to identify them on the basis of their characteristic. But here I have shown here this one of the type of rotifer. How you can identify the rotifer? Now, first of all, the posterior foot is having two toes. Entire body is what? Elongated. Broader at the center. This is general, not general characteristic, but specific of this type of a rotaria species. Okay, So, entire body is covered with a lorica. Lorica is a kind of a covering present around its body. So that you can see here, whatever structure is present is nothing but a lorica. It is a rigid or a semi-rigid covering present around the body of a rotifer. Right, posterior end is having two toes or forked tail-like structure is there, right, and that is again retractile. Whenever not in use or whenever it is disturbed, the animal is disturbed, the toes or posterior forked tail is withdrawn inside the body, right. So here you can see the brain, then stomach, then here is the anus, and what about this? This is a lorica, entire covering of the body. Okay, this rotifers, they are also called as a wheel animal killers. Okay, wheel animal killers. Why? Because of wheel-like structure, round moving wheel is the same way the wheel-like structure is present at their anterior side, right? So you can see here the structure, wheel-like structure, nothing but biforked lip with the cilia. So whenever the organism is in live condition, the cilia are showing a beating. So the beating of a cilia started from one end to another end. So it has a beautiful movement, and this beating of the cilia or ciliary movement gives a wheel-like, moving wheel-like appearance to the anterior end of the rotifer. So that is called, that's why it is called as a wheel animalculus, and this anterior portion bearing a wheel or a ciliary margin is called as a corona, and in the corona at the center point mouth is present, right. So if such a kind of a structure you can see, so indicate or considered is it as a rotifer, and the rotifers, they are free swimming and locomotion is takes place with the help of this toes. Okay, they again bear some kind of a leaping like movement with the, some are free swimming, some are use their toes for jumping or movement from one place to another place like this. Okay, after observing such an animal, when we change our field, right, when we change our field and we may get another type of organism, what is that? Say this, maybe you'll get this kind of a organism closely or having little modification, but overall structure is like this. So this is considered to be a Daphnia or Cladocera, right? Daphnia or Cladocera. Cladocera is a huge group under which Daphnia is one of the genus. Okay, but as Daphnia is most popularly observed, so here we have said the Daphnia, right? Example of the Cladocera is a Daphnia we have taken here. Okay, so how you will identify them? Okay, so if you see here again, you can see the outermost border, right? This clear-cut outermost border is there, but under which again something is there, right? Can you see this? Yes. So this outermost border is nothing but a covering, just like the lorica present in the rotifers. Here in this Cladocera or a Daphnia, what is there? A carapace. So outermost covering of the body, which is a hard, right, shell-like structure, that is called as a carapace. And inside that carapace, here you can see the legs are present. The carapace is closed from posterior side, but it is open on the ventral side, through which the anus comes out, as well as the legs, moving legs, they also comes out. So legs are present on the ventral side through the carapace. So carapace is open on the ventral side. It may carry egg pouches. So during breeding season, the Cladocera or a Daphnia carries egg pouches on their back. So you can see here the egg pouch or a brood chamber. It is also called as a brood chamber containing eggs, or you can say developing embryos. Right? So embryos are carrying, or brood pouches are present on the back of the Daphnia during breeding season. So this is also one of the characteristic feature, right? They shows a presence of well-developed compound eye. So here is the compound eye. So everybody is aware of the function of a compound eye. It is a sight organ of the vision, right? Now again after that, the antennae, two pairs of antennae are present, but out of which the second pair of antenna is very well developed and used for swimming, and a pair of abdominal setae are also characteristic feature. So not seen in the diagram, but here you can see like this, okay, like this. So huge number of setae, okay, here like structure present on the ventral side of the carapace or ventral side of the abdomen. This is again a characteristic feature that is called as a pecten, and it is again used or having multiple functions. So anal spine or posterior opening comes out to this, that is abdominal claw is called, and that abdominal claw is again visible under light microscope, which is a characteristic feature, right? So overall you can easily identify the Daphnia or a Cladocera under light microscope. And one more characteristic feature, their gut, right, their alimentary canal is very well visible in any condition at the light microscope. Okay, now let's move ahead. If you change your field, what can you see? Okay, now here comes the Paramecium. Okay, protozoa. Okay, so Paramecium is what? One of the protozoan organisms. And what are the protozoans? Protozoans are nothing but the unicellular eukaryotic organisms. Means what? They are having their entire body is made up of a single cell, and they are having usually one or two nuclei, and they are eukaryotic organism. Means they shows, eukaryotic means what? They shows presence of the cell organelle like membrane bounded vesicles, as well as the nucleus is well bounded in the nucleolar envelope, right, like that. Okay, so the protozoans, they shows a huge number of organisms are there, unicellular. Some of them, they may have presence of a chlorophyll pigment, and they can able to synthesize their food material. Some of them or most of them, they don't bear presence of a chlorophyll pigment, so they are not able to synthesize their food material, right? Okay, Volvox is there, Verticilla is there, right? So huge number, amoeba is also one of the protozoan animals. So huge number of protozoans are present in the water bodies. Now we have taken a representative member of the protozoa here. Suppose in the field, most commonly you will observe the Paramecium. Now how you will identify the Paramecium? Yes, you can easily identify it because of its slipper shape. So body is what? Body is slipper shaped, right? Body is having slipper shape. So this is the basic characteristic feature of the Paramecium, with the help of which we can easily identify it. Again, after that, you'll find the presence of a cilia. Huge number of cilia are present on their entire body. Right? Huge number of cilia are there. So entire body is covered with the cilia, and those cilia are quite larger at this end, oral end. Okay, or you can say at the tapering end. Okay. They shows presence of a two nuclei, one which is the larger one called as a vegetative macronucleus, and another one which is a smaller, that is a micronucleus. So this macro and micronucleus, that is two nuclei, is a characteristic feature of the Paramecium. Again, they shows presence of a two contractile vacuoles. So here a star-shaped contractile vacuole is present at one end, and at another end one more star-shaped contractile vacuole is also there, right? So what is the function of a contractile vacuole? Contractile vacuole involved in regulation of water, right? Osmoregulation is been carried out with the help of this contractile vacuole. So excess amount of water is being thrown outside the cytoplasm and maintain the homeostasis in case of this Paramecium or aquatic animal, especially protozoan. Okay, so here you can see the oral groove through which the food is taken inside, forms a food vacuole which is further digested, and defecated or undigested food material is thrown out through the anal pores. So this food vacuoles later on get fused with the anal pore, and through which it has been released outside. So that's all about the Paramecium. Let's see what is next. Next is Euglena, one more protozoan animal, unicellular, eukaryotic, means unicellular body is composed of a single cell. Then they are having number of cell organelle. So they considered as a eukaryotic. So they are unicellular eukaryotic organism. So it is nothing but a protozoan organism, though they shows presence of a chlorophyll pigment, but it is a moving animal, so it is a protozoan or a zooplankton. Okay. Alright. So Euglena, how you will identify the Euglena? Right? So you will identify the Euglena first of all with the help of their single long flagella. They bear a very long single flagella. With the help of that, you can easily identify the Euglena. And it bears, means its body is elongated, spindle shaped, broader, little broader in the center and tapering towards both the ends, right? And again, not very much tapering, it's a slightly, okay, as compared to the middle portion. Even if you see the structure of the Euglena, you can observe the chlorophyll granules present in it. So these are whatever greenish color structure is there, that is nothing but the chlorophyll granules. So with the help of the chlorophyll granules, it can use to, or it can able to synthesize its own food material, and hence it is not being taking any material from outside the cell as a part of a food material. It is synthesizing its own food material. It has one nucleus which is located usually in the center, guarded by the very well developed nuclear envelope, right? And it is also having a one contractile vacuole right here at the anterior point close to the flagella, base of the flagella. So there is presence of a one star-shaped contractile vacuole. So in the Paramecium, two contractile vacuoles were there, and in the Euglena, one contractile vacuole is there with a fixed shape that is present in the anterior portion close to the base of the flagella, right? Towards the another end, there is a no contractile vacuole, right? So this is a characteristic feature of the Euglena. You will write this in your journal. So while writing, so before that we'll go to the conclusion. What is the conclusion of this present experiment, right? Because conclusion is also important while performing any experiment. So as we are studying the pond water ecosystem, pond water sample we have taken for analysis. So what would be the conclusion? So the given water sample, that is given pond water sample, is rich in zooplankton. If you got some more, or you can say sufficient number of 2, 5, 4 or maybe the 10 number of a zooplankton accordingly in 3 or 1 to 2 drops. So you can say the zooplanktons or water sample is rich in a zooplankton. That is indication of a healthy pond ecosystem. So pond ecosystem is what? Functioning well, it is healthy. Right? If you got a water sample containing all the dead zooplankton, or there is no zooplankton, so what it means, if the planktons are primary producers, primary chain component of the food chain are not there, then that indicate higher, that is fishes, higher component of the food chain, that is fishes or larger animals, aquatic animals, they are also not seen, they are also not present in the ecosystem, right? So that is again kind of a sterile ecosystem. So if the planktons, zooplanktons are present in the sufficient number or in the optimum number in a given water sample, that indicates the healthy functioning ecosystem, right? Okay. Now dear students, after this, let's see what you will write in your journal. Focus here. First of all, this page, you will not going to write the first title page, right? Then see here, you will write this title. Don't write like this, whatever I have shown in the PPT, that is some shapes in which the words are there. No. What you will write it in a regular fashion. Only write this word introduction, and under which start from the next line pond ecosystem, that is a general introduction. Again leave one line, write requirement, and again start this. You will not going to circle the words as like shown in the PPT, right? Okay, you will write in a regular fashion. Then next slide is the pond, structure of a pond ecosystem. You will not going to write it. Understood? You will not going to draw this structure of a pond ecosystem. Okay, so don't draw this pond ecosystem. After that, this one, materials and methods, you will write this. Procedure also you will write in whatever given form. Don't make a circle to the words. After that, yes, on the drawing sheet, no need to draw this diagram. Okay, no need to draw this diagram. On the drawing sheet, you will draw this chart, flow chart of the procedure, right? You will draw the flow chart of the procedure. Don't draw this diagram. Okay. After that, go to the next page and write observations and results. You will write this entire thing and a rough diagram of the organism, whatever observed, or rough diagram of the zooplankton, whatever observed, right? So rough diagram with this. Don't write the web address, that is only HTTPS. This one you have not to write. You suppose not to write in the journal. Only the footer is given, that is diagram, under which write the name of that organism, name of that zooplankton, that is a cyclops, right? Like this, right? Okay. Now after that, the same thing here. Write the another zooplankton, rotifer. Write the characteristic and a small diagram of the rotifer with the footnote what is the diagram belongs to, and don't write this web address, or you can say image address from which the image is taken. No need to write in your journal, right? Okay, then next page, Daphnia, diagram draw properly. No need to write all the labels, right? Only give few labels like antenna, compound eyes, then brood chamber, carapace. This is sufficient, or anus, sufficient. No need to write all the labels, or no need to write give all the labels to the diagram. Again, don't write this image address. You are not supposed to write in your journal. Okay. Then this will also, you will write Paramecium with the characteristic and draw diagram of the protozoa, that is Paramecium, with the well labeled diagram. Write what diagram belongs to, and don't write this image address. Similarly, for this also you will do. Don't write the image address. Only here, after drawing the rough diagram of this euglena, you will write contractile vacuole, long flagella, nucleus, and a chloroplast or chlorophyll pigment. This 4 labels are sufficient. And then on the at the last you will write conclusion, right? So these are the things which you have to write, dear student. Okay.